Hello lovelies, in this video we're going to be going through your AQA AS Biology Paper 1, the paper that we've written for 2024. Now Dr Edwards is going to take you through all of the questions that she's written calmly and carefully, and what I want to do is pay attention to the exam skills as well as the content.
The image shows some cells from a garlic root tip viewed when using a light microscope. Give the name of the stage of cell division occurring in the two circled cells. Explain your answer. So they want us to say what stage of mitosis is happening and how do we know. So the phrase I always like to remember this is meet in the middle. And so if the chromosomes are in the middle or they're lined up in the middle, that is metaphase. Think meet, met, sounds the same. The other options, we can go through them, so it's not anaphase because you'd see arrow shapes, which you would see the chromosomes being pulled to the opposite ends of the cell, which we can't see. All those other cells that just have a circle shape in the middle, that is interphase. No chromosomes are visible, it's just a circle. That's also not a stage of cell division anyway, because that's a stage of before cell division happens. There are some prophase cells visible in this diagram where you can see that there's chromosomes starting to condense there in early prophase, so you can see that it's not just that blank circle where nothing's happening and it's all uncondensed. You can see that there's some kind of chromosome-like structures forming, so it's not that either. So we've ruled out pretty much all the others. It's very rare that you would get a telophase as a question because it's hard to tell that when that's happening. So it is metaphase. They're meeting in the middle, they're all lined up, they're kind of congregating around the equator, so that's how we know. So I'm going to put metaphase as our one mark, and then because the chromosomes are lined up along the equator. A student obtained the image after preparing a microscope slide. Explain a reason for each of the following steps in their method. So it wants the importance. Why did they do this at each stage of the method? What was the reason for it? So the first one is about cutting the first 5 millimeters of the tip of the garlic. And so hopefully we know that the reason we use the root tips is because that's where mitosis is happening. This question has been asked many times. It can be asked in slightly different ways each time, but really the reasons are there's a root tip involved. Why do we use the first few millimeters of that? Because that's where mitosis is happening. Why are they going to stain them? What's the purpose behind staining? Well, in this case, the whole point of the practical is to try and see the chromosomes. They would be invisible if we didn't stain them. So you can literally say that, or you can say the opposite way around and say to allow us to see the chromosomes so they can be visible. Why are they pressing down on it? Why are they squashing it? This practical, hopefully we recognise it as known as the root tip squash, and it's because you have to squash them. It's to produce a thin layer of cells, but that's not enough for the mark because that's not the why. Okay, so you're squashing it to produce a thin layer of cells because that means that light can pass through it, which you need to happen obviously in the light microscope. So to create a thin layer of cells so that light can pass through. Calculate the magnification of the image. Okay, so it's not asking us to measure anything specific. It wants us to just calculate the magnification, and obviously we can do that using the scale bar. So often you can get given information in the question in word form to figure out how to do this, or you're asked to measure the image size of something on the image. But this time we're just going to use the scale bar. That is something we can measure on the image, and it tells us the actual size underneath. So magnification equals the image over the actual size. Here's my ruler. I can show you what it looked like when I printed this off at home. And it's clearly 12 millimeters on my ruler. So you might have slight differences if you've printed it off at a different scale at home, but it's 12 millimeters. So once you've got that, we then obviously know the actual size is 20 micrometers because it's been put on the scale bar. So we can just sub in our numbers. So magnification equals the 12 millimeters divided by the 20 micrometers. But before we do that division, we have to convert. Always convert first, always measure in millimeters, and then the conversion to micrometers is easy because it's just times by 1,000. So we get 12,000 micrometers divided by 20 micrometers, which means that the image was magnified 600 times. The student calculated the mitotic index from their root tips. They obtained a different number to the students who had carried out the practical the week before with tips from a different garlic plant. Suggest 2 reasons for this difference. So they tell you they used a different plant. They could have done the practical at a different time of day, potentially. What else and what other things could affect the rate of mitosis, which is obviously linked to the mitotic index of the root tips? So it told you there was a different plant, so it could have been a different type of plant or have different genes. So it could have been a different type or variety or species of garlic, potentially. It would have had different genetics, potentially, if it was a completely different plant. It could be a different age, so the root tips could have been growing for a different amount of time by the time they measured it compared between the 2 plants. As we said, they could have done the practical at a different time of day, and that can make a difference to the rate of growth because they'll grow at different times of the day, especially depending on light levels. So there's also other growth conditions if you think about the fact that garlics could be grown differently, so anything that could affect growth rate, you can use some of those kind of like abiotic conditions as well. You just need 2 of these. And this is like something, if you think about, most of the time we talk about when we do practicals with plants, we say get the same type of plant or take your sample from the same tissue, because obviously there can be differences. And in this case, genetic differences or anything that can change the rate of cell division is important and obviously could change our results. So the garlic could have been a different variety. That is the same thing as saying a different type of garlic. And the root tip could have been growing for longer. So the age of the garlic could have been different. The image shows a blood smear viewed under a light microscope. Give the letter that represents a phagocyte. So hopefully we know that's a type of white blood cell. The majority of like the disc shapes in this image are going to be red blood cells. The kind of little scraps labelled Z are going to be platelets potentially. And then we've got X, and then we've got a cell that's not labelled. So the cell that's not labelled is a lymphocyte, and then the cell that's labelled X is a phagocyte. So that is a white blood cell. It's bigger, it's larger, it's got that multi-lobed kind of nucleus, the dark kind of sort of joined string of beads shaped nucleus, and it's very clearly not a red blood cell or a platelet, so it is X. A biologist is studying the concentration of red blood cells in a 10 milliliter sample to assess the health of an individual. To do this, they used a hematocytometer grid, viewed under a microscope. The grid is divided into multiple squares, each with a known volume. The volume of one large grid square on the hematocytometer is 0.1 millimeters cubed. The biologist dilutes the original blood sample with a saline solution in a 1:20 ratio. Using the hematocytometer, they count the number of red blood cells in 5 of the large grid squares. The average number of blood cells counted in 5 squares was 8.18. So remember, one large grid square is 0.1 millimeters cubed. Explain why the biologist had to dilute the blood sample. So this is a similar question that you might get if you're sort of talking about diluting a bacterial broth to count the number of bacteria in it as well. Anything like this, we've got like lots of floating around cells or substances in there and we want to try and count them. Diluting them makes it easier to see or easier to count them, because there's need to not be too many of them in the grid. If there's loads, it's all crowded, you can't tell one individual bacteria or one individual red blood cell from another one. How can you actually see them to count? So by diluting it, we make it easier to count them, easier to see. Calculate the total number of red blood cells in the sample. Give your answer in standard form to 3 significant figures. Okay, so we need a total number in the sample. The sample is 10 millilitres, remember? And we've managed to do a sample. We've got an average of 8.18, which is 8.18 on average in 0.1 millimeters cubed of the diluted solution. And we've got a dilution factor of 1 in 20. So we need to multiply the average by the dilution factor, or multiply the average by 0.1 millimeters cubed, then multiply that by the dilution factor, and then multiply that up to get the total volume of the sample. So 8.18 times 0.1 millimetres cubed means we have 81.8 red blood cells per millimetres cubed. Then we're multiplying by the dilution factor, which is 20. That gives us 1,636 cells per millimetres cubed of sample. And there's 1,000 millimetres cubed in a milliliter. So 1,636 times 10,000 millimetres cubed gives us the final answer, which is 16,360,000. We need to put that in standard form to 3 significant figures. So we're going to do 1.64 because we can round up that 3 because of the 6. And then it's times 10 to the 7. If you're always unsure about the jumps, I do the little jumpy thing to make sure I'm happy with the decimal point. So where the decimal point is in your standard form to the end of the number is 7 jumps. That means it's 10 to the 7. Cells being infected with a virus called HPV can increase the risk of getting cervical cancer. HPV is spread from touching infected skin of another person and can be a sexually transmitted infection. A vaccine has been produced against the HPV virus and is offered to 12 to 18-year-olds by the NHS in the UK. Explain the role of T cells in the body's response if a vaccinated person becomes infected with HPV. So we're talking about someone who has had the vaccine, so is potentially immune, and we're just focusing on the role of T cells. So we don't need to explain the whole sequence of what will happen. We're just focusing on what the T cells' role will be. So what do they do when someone is infected with the pathogen for HPV? But in this case, it's the secondary immune response because they will have already been exposed the first time when they had the vaccine. So we're used to talking about memory cells. So there are T memory cells, which are going to recognize the antigen. They're going to stimulate the B cells, and they're also going to stimulate like T killer cells and things like that. So it's this time, because it's secondary immunity, we don't go through the whole process of activating T cells and B cells. We just activate memory T cells and B cells that will remember this pathogen from before, and so they quickly go through division by mitosis to make loads of themselves in order to start the immune response straight away. So the memory T cells are activated by the HPV antigen. They divide rapidly by mitosis. That's going to produce the T helper cells, which activate B cells. You've got other options in the mark scheme in terms of the other cells that can be activated that can then trigger more of the immune response. The graph shows the percentage of the population of 12 to 18-year-olds who have been vaccinated against HPV and the number of cervical cancer diagnoses per 100,000 of the population since the year 2000. So although this graph looks quite like a lot, you've just got two different sets of data that are related to each other in a way, or they think there's some correlation in a way, and so they've plotted them together on the same graph so you can see how the patterns relate to each other. Scientists have concluded that there is a direct correlation between introducing the vaccine and a decrease in the number of cervical cancer cases in the UK. Evaluate the scientist's conclusion based on the data from the graph to support your answer. So this is an evaluate question. Okay, so we're basically asking, does the graph and the data in the graph support their conclusion that it's the vaccine that's led to the decrease in the number of cervical cancer cases in the UK? You have to do both sides. So yes, does it support, and how can we see that? And also maybe no, why does it not support that, or what other things could be able to explain this correlation? So in terms of supporting, obviously as the number of 12 to 18-year-olds that have been vaccinated against HPV increases, then we can see that the number of cervical cancer cases reported has decreased. And it's continued to decrease. Even though it's slowed down, it's still decreasing all the way to 2019. However, the decrease and the steep decrease in cervical cancer cases started in 2004, before the vaccine was used, and that steep decrease is happening when there's only kind of a few, like 10 to 15% of the population vaccinated in terms of the group of 12 to 18-year-olds. So that decrease is probably not being caused by the vaccine at that point. And if you look at when we get to a higher increase in the number of people vaccinated, the actual number of cervical cancer cases slows down, like the decrease slows down, which you'd expect it would be even steeper if the kind of vaccines were causing that. So there's probably something else going on there. It's obviously the vaccine will have contributed, and we can explain why having the vaccine makes you immune, and that's a good thing, and that would potentially explain why that would decrease the number of cervical cancer cases. But the evidence of it decreasing before the vaccine started, and when there's very few children who are vaccinated, that steep decrease, there's probably other factors at play here. So it may not be the only reason. And this is kind of this idea of cause and versus correlation. And there is a correlation, and it's probably part of the reason why there's a decrease, but it's probably not the only reason, and there could be some other things. And when there's a disease or the prevalence of a disease decreases over time, there's often a lot of factors at play. So things like increased awareness. So more people are aware of what might cause them to get this disease, and therefore they will change their lifestyle, change their habitats, or they will change how they go and get tested to make sure that they are not unknowingly then passing it on to other people. Things like HIV, you can use the similar arguments as to why HIV cases have reduced over time, and partly it's because we have new medication to help stop prevent the spread, but also things like condoms are more freely available, things like the fact that people are more aware of HIV and how it gets transmitted, so they will take steps to prevent that. And that's happened all in the years since we've sort of researched HIV and know more about it. And this is potentially a similar case here. So you don't always have to say that every bit of data supports. If you can find reasons in the data to show why that might not be the case, and use some of your own knowledge as well, because that's absolutely fine, especially when you're kind of speculating why something might not be completely the reason that something has happened, you can definitely use your own knowledge here. So I'm going to go through my answer, and kind of you can see where I've picked up the marks, and I've tried to make sure I've talked about data from the graph and talk about both sides of the argument. So the graph does show that as the percentage of the population vaccinated against HPV increased from 2005, the number of cervical cancer cases has fallen. However, there was a decrease from 2004 to 2005 before the vaccination programme began, and the sharpest decrease in cancer cases occurred when very few children were vaccinated. So I've got kind of the pro and con of the decrease and the increase pattern, so there are two sides there in one paragraph. Being immune to HPV would reduce the spread of the virus and therefore reduce the chance of cervical cancer in these vaccinated people. And although the decrease in the number of cases is slowing down, it could just be a delay in seeing the effect, as children are vaccinated but adults are most likely to be diagnosed with cancer. So again, I've kind of used my scientific knowledge to show why and explain why the increase in vaccinating people would prevent the chance or reduce the chance of them getting cervical cancer. And that could explain the decrease, but also there could be a reduced decrease and slow down in the number of cases that have been found, partly because we're vaccinating children, and most of the people who are being diagnosed with cervical cancer are adults. They're going to be older than 12. So there's potentially a delay in us seeing that change and the change the vaccines are having on the number of cases of HPV. This suggests that there could be other factors contributing to the decrease in cancer cases. Since 2004, there could have been an increased awareness of the link between HPV and cervical cancer, so more people will try to protect themselves from infection or get regular tests to check for infection. And that means that they obviously wouldn't then pass it on as well. So I've then, sort of my last paragraph is saying that although the data has shown some evidence, there's also some evidence against supporting it, and therefore there could be other reasons, and these are my other reasons. You don't even have to go to that final paragraph in your answer to get the full 5 marks, because we would have got, or it's easy to get 5 marks just from talking about the data, just from giving some numbers. I didn't actually provide any percentages or any sort of difference in numbers or calculation to show how the data could actually support the points that I've made. So you can get extra marks for that, and you need to have some discussion points that show that it might not support the conclusion, but there's obviously lots there that you can say, and there's so many marks available in that mark scheme. One thing we didn't talk about was obviously statistics. So obviously, if you were going to show or trying to show that there's a direct correlation occurring, then you need to make sure that you actually plot the two variables against each other like a scatter plot, and then you could do a statistical test. You could use the correlation coefficient to show if there's a significant relationship or not. So that is something that we could have talked about that they could have done to actually show that direct correlation. The diagram shows some chromosomes in the nucleus of a cell. Draw around one pair of homologous chromosomes in the diagram. So, homologous chromosomes should be the same size and shape because they're the same chromosome, just one of them came from each gamete or each parent. So, the easiest one here is to find two that look very similar in size. I'm going to pick the two smallest ones. That seems the most logical to me. If you were picking some of the others, you've got options for a large one. There's two large ones, there's two medium ones, there's two small ones. So as long as you put a ring around two that match, so in my case I did the two small ones, that will get you the mark. Explain what features would make a pair of chromosomes homologous. So features often thinks about structural or physical features. We've already said it's got to be something to do with the same size or shape that we've seen, and that's how we've identified the one for part A. But we know that what it's basically asking us to define what a homologous chromosome is with some three things that you can say that homologous chromosomes are, what makes them homologous. So they are the same size and shape, they are the same chromosome but one from each gamete or one from each parent, and they will contain the same genes in the same locations. They can contain different alleles though, and obviously that means that they are not identical. So you don't get any marks for talking about them being identical because they're not, because one comes from each parent. So although they will contain the same genes, they can contain different alleles. You've got loads of things you can say for these options in the mark scheme. I'm going to go for the fact that one chromosome came from each gamete or each parent. They're going to contain the same genes in the same order, and they're going to be roughly the same or similar size. Identify what is the diploid number of chromosomes of the organism from which this cell was taken. Diploid meaning the full number of chromosomes. So this cell was taken from an organism. We're just being asked to say what the diploid number of chromosome is of that organism based on what this cell tells us. So there are 6 chromosomes, 3 pairs of homologous chromosomes, 6 chromosomes in total, so our diploid number is 6. Can't just say 3 pairs because it asks for the actual number. Draw a diagram in the circle below to represent the chromosomes that are present in the nucleus of a gamete from this organism. So this time it wants us to represent a haploid cell. So half the number of chromosomes, so it will have to be 3 in total. And at the end of meiosis, we have chromatids, okay, or individual chromosomes, but it's made of one chromatid from each homologous pair. So that means we need one large chromatid, one medium chromatid, and one small chromatid. So we're just doing one of each. So one chromatid from one chromosome of the homologous pair, because that's what happens. So in the first stage of meiosis, we separate the homologous pairs of chromosomes, and in the second stage of meiosis, we separate the chromatids from those, the sister chromatids of those chromosomes. So we end up with one chromatid from each chromosome from each homologous pair in each of the 4 gametes. So we have to make sure we know that there's 3 different chromosomes, so we've represented that with the 3 different lengths of chromatid. And you can do that with or without the centromere, that's fine. As single chromatids, they aren't always represented with the centromere, so you can do it with or without. With things like this, you just have to make your drawings really clear. So it doesn't really matter what the length is, as long as you've got one small, one medium, one large, and you can really clearly tell the difference that you've got 3 different lengths of chromosome there. After carrying out a heart dissection, students wanted to investigate the physical properties of arteries and veins. They cut rings of artery and vein tissue and then set up some equipment as shown in the diagram. So we've got a hook, we've got a ring of artery or vein tissue, and it's been attached to a mass carrier and some masses have been added. And we've got a ruler behind it so we can measure the length of the ring of tissue. When it's talking about measuring the physical properties of arteries and veins, this is going to be about strength, elasticity, so the different structures enable them to have those different properties that we know arteries and veins have. so their structure matches their function. This is to look and investigate and kind of compare the differences between those two tissues. Give 2 safety precautions that students must use when collecting and handling the vessel material. Related to dissection, and the skills we've got here is thinking like handling biological material, and then also thinking about kind of cutting, and actually it says collecting and handling. So we've got this idea of what should you do when you're handling this tissue, how should you be safe, but also when you're collecting it, so they must have cut the rings of tissue, it says in the start of the question. So our cutting skills, scalpel use skills, how to be safe when we do that. So the main 2 that spring to mind for me here was to wash your hands after you've touched and handled the material is a safety point. Cutting safely, so cutting away from you or cutting onto an appropriate surface, not into your hand, things like that. Or the idea of kind of disposal of the material or cleaning up and things after using, obviously, material that are potentially a biohazard. So any of those, and you've got the mark scheme to kind of give you the more detail that you can pick. We've just got to pick 2. So washing hands after handling the tissue is going to be my first one. Again, you have to be specifying that it's after handling the tissue. You can't just put wash your hands. That's not an effective and descriptive enough answer to get the mark here. And then cutting away from the body when slicing or cutting the vessel tissue. The results are shown in the table below. So we've got some rows for the artery, we've got some rows for the vein. The masses added were the same for each one. We've got the original length with the mass carrier, and then we've got the new length with the masses. So every time we added another mass, we'd measure it with the mass carrier, and then once the masses were added. Calculate the difference in percentage length increase of the original piece of tissue between the artery and the vein by the end of the experiment. So it wants us to calculate the percentage length increase from the start to the finish of the experiment for the artery and the vein. So from 4 to 5.2 centimetres for the artery and 4.5 to 5.1 centimetres for the vein. Calculate that percentage length increase and then calculate the difference in that increase between the artery and the vein. So percentage increase is going to be the increase in length divided by the start length times 100. So for the artery, that's 5.2, because that's the final one, minus 4, which gives us the difference or the increase, divided by 4 times 100 gives us 30% increase in length. And then for the vein, it's 5.1 minus the 4.5 divided by 4.5 times 100 gives us 8.51% increase in length. So the vein obviously increased in length less than the artery. So the difference between the 2 percentage increases is 21.49%. And if you round it up to 21.5, then you still get the mark here. Explain the difference in the results between the artery and the vein. So it's asking us to give reasons for the difference in the results, i.e. the difference in the change in length, because we have just calculated that. But you can also get other things from the results table you could use for this as well. So my main thing I'm trying to think of a reason for is, why does the artery stretch more? Why is it an extended length as masses are added? Why does it stretch more with the same amount of mass? Well, what can I link to the structure of arteries? The arteries are more elastic, right? This shows they must be more stretched, able to be stretched more, so they have more elastic tissue. We should know that. So then we need to link the idea of, why do they have more elastic tissue? Well, this is because they need to stretch and recoil to maintain high blood pressure. So this is really a structure and function question about arteries. It's shown you that they've stretched more, they've got longer when the same amount of mass is added. You're trying to come up with a reason why. So the difference between arteries and veins is that the arteries stretch longer. The physical difference between arteries and veins is that arteries have more elastic tissue. The reason arteries have more elastic tissue is because they have to stretch and recoil to maintain high blood pressure, which obviously veins don't need to do. The artery stretched more than the vein because it contains more elastic tissue. So with one sentence there, I've kind of got 2 marks because I've talked about the difference that we've seen. and then I've given a reason, one reason for the difference, and then I've got to say why. This is because the artery has to stretch and recoil to help maintain high blood pressure. The graph shows the result of an enzyme-controlled reaction repeated at 3 different temperatures. The volume of product formed was measured every 2 minutes for 60 minutes for each reaction. Explain the difference between the curves at 37 and 60 degrees. So we need a reason why for the differences in the rate or the patterns that we can see. So there's a steeper initial rate of reaction for the 60-degree temperature, and then it flatlines, but it flatlines pretty early, much earlier than the 37 degrees. There is also not as much product, so the volume of product made was not as high for the 60 degrees compared to the 37. So the suggested reasons then are obviously that 37 degrees is the optimum, and if we think about the differences between that and 60 degrees, it must mean that at the 60 degrees there was a denaturation of the enzyme, which is what caused that early flatline. But the initial rate of reaction was faster in the 60 degrees because there would have been a higher temperature, so therefore there'd be more kinetic energy, so more successful collisions made between the enzymes and more enzyme substrate complexes made, and therefore product formed quicker in the first few minutes. But then there's a flat line which suggests the enzyme was denatured, which means that the active site's changed shape, so it can't bind to the substrate anymore, so not as much product was formed. So at 60 degrees, there's a higher initial rate of reaction, down 37, as there is more kinetic energy, so more enzyme substrate complexes are formed. As long as you say things like higher, you don't have to specify like explicitly 60 degrees is higher than 37, and therefore this happened more at 60 than 37. If you're saying higher or more than, so you're kind of comparing it in that language, that's fine. So at 60 degrees, the enzyme's denatured, so less product is formed than at 37 degrees. Explain why less product was formed at 25 degrees compared to at 37 degrees. So this is a similar thing now, we're just comparing 25 to 37. Different shapes on these graphs though, because obviously we've got the higher rate of reaction for 37 than a flat line, and we don't have that for the 25 degrees. So this time we're going to say the opposite. There was a lower temperature, so there's less kinetic energy. So there's a lower rate of reaction, which explains why that line is like that for 25 degrees. If you were to keep extending that line, if there was more time, that would keep going probably in that rate until it reached the same amount of product as the 37 degrees. It just is going to take a longer time to get there. And we don't have that because all of them were ran for 60 minutes. And this is because at that point, there's nothing to do with denaturing the enzyme. It's just that not all the substrate has reacted yet. And it will, given time with that enzyme, it will just take longer because there's less kinetic energy. So at 25 degrees, there's less kinetic energy, so there are fewer enzyme substrate complexes formed. This means the rate of reaction is slower. This means that not all of the substrate has reacted after the 60 minutes. The experiment was repeated, but with an inhibitor of the enzyme. A graph of substrate concentration against rate of reaction was plotted. It looked similar to the line produced at 60 degrees. Okay, so this time they are repeating it, as in they're repeating an experiment with the enzyme, but this time they're doing it, instead of changing the temperature, they've added an inhibitor. And so what they've done is they've then plotted this little graph. So it's not the same graph because that's time versus volume of product. The graph they've said they've drawn for this one is substrate concentration and rate. And they said the line looks similar to the 60-degree line. So that means it goes up and then it flatlines. So they want you to be able to explain from this evidence that they've given you in the question what type of inhibitor was used. So you've got a couple of clues that we can get from this question, and I would absolutely have sketched out this graph because having a visual is really going to help. So when it says type of inhibitor, we have to choose, is it competitive or non-competitive? And you need to be able to explain your reasons why you've chosen it, and that idea that you've got this shaped graph at 60 that looks similar to the 60-degree shape, that's kind of your evidence, isn't it? So there's a couple of things you can get from this. High initial rate of reaction, then a flat line. So as the substrate concentration is increasing, the rate of reaction isn't increasing. Therefore, there's got to be something else stopping it, or it's behaving almost as if it has been denatured, because that suggests that there's initially a rate of reaction, there's enzyme substrate complexes forming, and then it stops. So when all of that inhibitor is bound to all of the enzyme, it changes the shape of the active site, so it can no longer bind to the substrate. So effectively, if you understood that at 60 degrees, the shape of that graph was caused by the enzyme denaturing, you can link that to this idea that you know it must be a non-competitive inhibitor, because that would be the one that would change the active site shape. A competitive inhibitor wouldn't do that, because it would be a slower rate of reaction. It would look similar to the 25-degree line, for example, because it would just be a slower rate of reaction, but eventually it could get there. So you can pick which bit of evidence you want to use. There's lots of different ones in the mark scheme. I'm going to go with the kind of relating it to like it's being denatured. So it's a non-competitive inhibitor because the active site of the enzyme changed shape so that the substrate can no longer bind. Tuberculosis, or TB, is a type of lung disease caused by a bacterial infection. The infection destroys patients' lung tissue. Infection with TB can have an effect on lung volume. Describe the process of exhalation. Okay, so exhalation is breathing out, and describe is obviously just steps in the process of how this happens. It's only 3 marks, so it's not super long, super detailed. We just need to give kind of like the steps that cause air to leave the lungs. So your diaphragm relaxes and your lung volume reduces because your ribs move in, your rib cages move in and down. So the reduced volume creates higher pressure inside the lungs, and then there's lower pressure outside the lungs, and so that difference in pressure, the gradient, forces air out, down the pressure gradient. So the diaphragm relaxes and moves up. The ribs move in and down, reducing the volume in the lungs. The pressure in the lungs becomes higher than external pressure, so air is forced out. A spirometer can be used to measure the forced vital capacity, or FVC, tidal volume, and breathing rate. Spirometer measurements of FVC taken from patients who have multiple infections with TB can be seen in the graph below. So we've got the mean volume of air that was breathed out, and we've got their age groups in years, and then how many infections they had, either before they had any and then after their first and second infection. Describe what the graph shows about the effect of TB infection on the FVC. So there's a consistent pattern here that more TB infections equal a reduced forced vital capacity, or that's the measure of volume of air that the lungs can breathe out after one big intake of breath. This suggests that kind of TB infection reduces lung volume each time you have an infection. And because it's a describe question and it's asking you to show what the graph is telling you, we're going to need to put some data in our answer at some point. So I'm just going to pick sort of one group of columns and talk about the difference from before TB to after TB. You could calculate that as a percentage change if you wanted to. You could just talk about the difference, which is what I'm going to do. So it's 4.1 to 3.3? 3.7, because that's 3.5. So 4.1 to 3.7. So that's the difference. I'm going to talk about that as my data point. There's other data points, obviously, you could talk about. The one thing that we don't need to talk about here, though, is age, because obviously lung volume decreases and with age, but the effect of the TB is the same across all the age groups, and that's really what we're being asked to talk about here, is the effect of TB. So TB reduces lung volume with just one infection, but the more infections with TB, the more the FVC is reduced. For example, in the 35 to 39 year group, it reduced the volume by 0.12 with one infection, but 0.4 with 2 infections. The damage from a TB infection creates a thick layer of scar tissue around the alveoli and reduces lung elasticity. Explain how a reduction in lung elasticity can make it feel harder to breathe. So that means there's an elastic layer around the alveoli. This is the tissue that we're talking about. And obviously that allows us to inflate the lungs when we breathe in And then there's that recoil element as you exhale that helps to force air back out of the lungs. So because it's asking about why it's harder to breathe, it doesn't say breathe in or out, so it's suggesting about breathing in and out. So it's about inhaling and exhaling. So for the first mark, I'm going to say that the lungs will not inflate as easily, so breathing in will be found harder. The alveoli will not recoil when deflating, so breathing out will feel harder or more effort will be needed. So you'd have to kind of force it, which is why they've got that mark in the mark scheme for it not being as passive. Explain how the damage to the alveoli walls will reduce the efficiency of gas exchange in the lungs. Anytime we're talking about efficiency when it comes to gas exchange, it's really talking about the adaptations that that exchange system has, normally to maximise diffusion of gases. Something's obviously stopping this or preventing it or reversing it. So we've got the scar tissue, it says there's a thick layer, so that means we've no longer got these super thin alveoli walls, which help to reduce the diffusion distance. The walls of the alveoli suffering damage also is going to reduce surface area. So we know that we've got these kind of shaped structures that create this large surface area. If we damage that, any damage to alveoli in a question is often referring to the fact that it's reducing the surface area of the lungs. Alveoli walls are thickened by scar tissue, so the diffusion distance is increased, and damage to them reduces the surface area available for gas exchange. So I've got my keywords in there about kind of the diffusion distance, the fact that surface area is reducing, and how that's going to reduce the gas exchange rate. Human activity is impacting biodiversity. Peat bogs are unique wetland habitats for many species. Peat bogs are being drained for human use, including grazing of livestock. Peat can also be cut into blocks and extracted for use, as seen in the image below. Explain what is meant by the term biodiversity. Explain what is meant by the term questions are just define questions. So biodiversity is described as the variety of different species living in an area. And you've got various options for talking about area, habitat, ecosystem. As long as you're talking about the fact that you're talking about the number of different or the variety of different species, that's what we mean by biodiversity. Explain how human activity has reduced biodiversity in the peat bogs. So it gives us some clues in the question. It's talked about the fact that they've drained it, so they've removed the kind of wetland element, they've drained the water, they've got animals eating the plants through grazing, they're also removing the peat itself. So this question has got some clues in the question to help you, and also we may remember about peat from GCSE, but it's about making sure that you're explaining why those things are reducing biodiversity specifically. So reducing biodiversity means we're reducing the number of different species or organisms in that peatland area. So we've got always the same answer. If something's talking about why is biodiversity being reduced, we've got the same few things we can talk about. There's fewer habitats, there's fewer niches, there's fewer food sources. And you have to say food sources, not just there's less food, because it's not about the amount of food of a single species overall. It's about that there's fewer different options of species that they can use for food, and that reduces therefore the number of organisms that eat that food or rely on that food. So there's fewer food sources, and we can link that to the explanation of because plants are being removed because of the grazing animals. We can talk about the fact that there's fewer habitat, or the habitat has changed because they've reduced or got rid of the wetland element and they've dried it out. So that means that species are adapted to live there can no longer live there. There's lots of ways we can explain this, and you've got lots of different options in the mark scheme. I'm going to go with fewer plant species due to grazing or extraction. So we've literally removed plant species in that way. And that also means there's fewer food sources. The draining of the wetland also reduces the unique habitat, so specialised species can no longer survive. Okay, so I've actually probably done more than 3 marks worth potentially, because I've got fewer plant species, fewer food sources, and the changing of the habitat, but that is 3 marks. And you can obviously talk about more things, but this is the kind of standard answer that we would normally try and apply to a biodiversity reduction question. Give 2 reasons why biodiversity should be maintained. So why should we maintain biodiversity? It also goes for conserving it, protecting it. Why should we try to keep high biodiversity? Why is high biodiversity important? So there's a few reasons we should maintain biodiversity. The first one mainly is the moral or ethical reason that we shouldn't just let species go extinct as a result of human activity. That's not okay. We need to safeguard those species for future generations. Those species in high biodiverse areas could provide resources for us, medicines for the future that we haven't yet discovered, other resources that we use plants and other organisms for, and also potentially like we make money off of them through trade or ecotourism, so there's a financial incentive as well there. There's lots of reasons in this mark scheme and kind of the way you can word it, but I'm going to go with sort of two about the kind of moral, ethical, and the resources. So biodiverse habitats could be a source of future medicines for humans. We have a moral duty to prevent species from going extinct. Read the following information. So this is our comprehension style question. That's always at the end of this paper. So we're used to seeing these kind of long answers, and there's going to be lots of questions about different topics from across the spec, different skills. And there's sometimes clues in the question, and sometimes it's just we have to use our knowledge. So first up, it says explain how water and ions are transported in the vascular tissue from the roots to the leaves, lines 3 to 4. So if we go and find where that is, highlight that. Once inside the plants, the ions can travel through the vascular tissue into the leaves where photosynthesis can be disrupted. So really, this is an explain how water moves, what's the process that water moves up through the xylem. It doesn't mention the xylem, it says vascular tissue. You obviously have to know that if it's water and mineral ions travelling, then it's xylem. And so really this is just your knowledge about the cohesion tension theory. So transpiration occurs at the leaves. This lowers the water potential in the leaf tissue, which creates tension. It's the tension that pulls water up the xylem, or it creates that negative pressure, which pulls water up the xylem. And the water molecules, as they move up through the xylem, are cohesive, so they form this unbroken column of water. So there's many ways you can be asked this question. This is a 3-mark version of this answer. You could give lots more detail and go into a lot more description and explanation of how the cohesion tension theory works, but this is enough just for this 3-marker that we've got here. Explain why having a high concentration of ions in the soil reduces a plant's ability to absorb water through its roots, lines 5 to 6. So it says salt concentrations dissolved in soil water make it, or high salt concentrations make it difficult for plants to absorb water. This increases water stress and root dehydration. So again, it's not told you anything necessarily extra or different in that statement in the comprehension. This is just being able to give reasons why having a high concentration of ions outside of the root cells in the soil around them might reduce or prevent them from being able to absorb water. This is just an osmosis question because we should understand and know that plants absorb water from the soil into their roots by osmosis. So we should be able to understand that if there is a low water potential around the root cells outside, because there's lots of ions dissolved in that water, then if the water potential inside the cells is higher, then water is not going to move into the root cells by osmosis. If anything, it could move out. So they could lose water and become dehydrated, or they're going to find it very difficult because you can't actively transport water into cells. You have to rely on it going down its water potential gradient. And so if that's not going to happen, then you're not going to get as much water into the cells. High ion concentration in the soil lowers the water potential around the roots. If the water potential of the cells in the root is higher than the soil, then the water will not move into the roots by osmosis. The main thing is we've talked about water potential here, where it's higher, where it's lower, where water will or won't be moving. And we've mentioned the word osmosis, which is a key word. Have to have that in our answer to get that mark. Describe 2 ways the genetic diversity within a crop species could be analysed. Lines 11 to 12. It says the need for development of a salt stress resistant crop variety is high, but given the limited range of genetic diversity in current crop plant species, scientists are looking into other tolerant species. Okay, so this is just telling you that diversity in crop plants is low in terms of genetic diversity. The question is just describe 2 ways that genetic diversity could be analysed. So how do we identify differences in genes between organisms. Hopefully we've seen this question many times before, and the mark scheme is always the same. If it's asking about how would you be able to tell whether two species are genetically similar or not, or more closely related to each other than not, or how diverse and how many differences there are in their genomes or whatever between them, the answers are always that you're going to compare the DNA sequences, you could compare the RNA sequences, or you could compare the amino acid sequences. And this is what you always get marks for. The key thing here is to talk about the base sequence of DNA and RNA, and then the sequence of amino acids. So I've put compare the sequence of DNA or RNA bases, and that will get me 2 marks. If you think you'll forget the word bases, put compare the base sequence of DNA or RNA, and then you make sure you put base sequence as a whole phrase, and that means you'll always get the mark for that. Root tips are softer and more flexible than older root tissue closer to the stem of the plant. Suggest why. Lines 18 to 19. So it says, enables plants to provide protection against salt stress for sensitive cells in the root meristems. These immature cells do not contain a vacuole, which can usually help to store or dispose of harmful substances. So this is a suggest question, which means there'll be options in the mark scheme for what you can say, because it's using kind of your own knowledge, or in this case, knowledge from the text. So there was a clue in this phrase where it says there's no vacuole. So if there's no vacuole, then they're going to have reduced kind of turgor pressure inside the cell. The vacuole helps to swell with water and make those cells turgid, which makes them stiff and rigid. And that's what helps give the support to the plant. So there's a clue there in the question that suggests and can tell you some information. This is not always the case with these comprehension questions, but with suggest questions, there's often some clues if it's going to be having to be from your own knowledge or something that you can think is common sense. As you can see the mark scheme, there's other options there. So thinking about root tips might make you think, okay, that's where mitosis is happening. So they're flexible, so they can grow and bend, down with gravity or whatever. You might have thought about that, or you might have thought about lignin, which we know is a structural support material that isn't found in those kind of root tips. But the clue is in the question there, so that's the answer I've gone for. So the root tip cells do not have a vacuole, so they have reduced turgor pressure. Describe how the protein found in Arabidopsis transports ions out of the cell. During salt stress, a transport protein is activated which pumps sodium ions out of the cell and protons into the cell. So how is it moving the ions? What process is it moving the ions by? That's a kind of, give me a description of how these ions are moving using this transport protein. It says pumps. There's a clue in the question. So that's suggesting to us that it's active. It's obviously potentially moving sodium ions against their concentration gradient, because if the sodium ions have come in because there's more sodium ions around because of the salt stress, then it's actively trying to get them out. They'll be moving them against their concentration gradient. And it mentions this idea of sodium ions out and protons in. So this could be a co-transport setup. It's going to be some kind of carrier protein that's physically moving ions across the membrane. And we've mentioned active transport, so that's a lot there. So you can either go down the active transport route or the co-transport route, or both. And you've got to get this idea that it's pumping, or it's actively moving ions, and it's moving some of them, sodium ions potentially, against their concentration gradient. So active transport using a carrier protein to move sodium ions against their concentration gradient. Describe how the ribosome in the meristem cells produce the transport protein. So without even needing to go and look at the line, this is definitely just a translation question for 3 marks. How does a ribosome make a protein? But it says the formation of this protein is increased in root meristem cells. So it's only 3 marks. It's not going to be sort of 5-6 mark levels of detail, but we need to talk about the mRNA, we need to talk about tRNA and binding to anticodons, we need to talk about amino acids being joined together to form a polypeptide with a peptide bond. And so they're my kind of 3 go-tos that I would definitely mention to make sure I get the marks. With something like this, if it's 3 marks, it's potentially worth writing a little bit more. And if you've got a kind of bullet point process for translation with all the keywords and all the detail in that you've learned, and you can just quickly rattle it off, then by all means, then do that. even for a 3-mark question, because it just guarantees you get those marks as long as you're not spending ages thinking about the whole process. So I've got the mRNA binds to the ribosome to be translated. I've mentioned the word translation because there's no mention of that in the question or the text, so I know I'm probably going to get a mark for that here. tRNA with the complementary anticodon binds to the codons. You could say multiple tRNAs if you wanted to. The ribosome then catalyzes the formation of a peptide bond between the amino acids. On each tRNA, the ribosome moves along to the next codon. So I've put at least 4 points in here. I might have even got 5 marks worth in here, but that's fine. I didn't spend too long thinking about it. It's kind of a sequence of bullet points or statements that I know quite well how to describe the process of translation. I've mentioned my keywords like anticodons and translation in there to make sure I've got them. If you found this hard, 1, interpreting that it was about translation, and 2, not just knowing sequence of steps of translation off by heart, then use that mark scheme that we've got. Try and make sure that you can memorise that for the next time this comes around.
Ouch! This is why in some videos I explain scratches.